So what does pharma look like in 2040 and 2050 and 2060? We had this huge rush in the 90s. That was kind of the pharma small molecule glory days. And in the background, biotech was having its own little, coming out party. And then antibodies kind of took over for the 2000 to present day, basically, era. But now what? The multiple myeloma had no FDA-approved drugs. And the IMiDs came around. Proteasome inhibitors were the first approvals, and we don't even use them that much anymore. They're not as potent as a CD38, CAR-T. You got 20, 30 choices for multiple myeloma. And, a lot of people talk about post-finasteride syndrome, fluoroquinolones as well. A lot of people think if you take, the azoles for fungal infection, that you have this long-term side effect package too. So small molecules are just kind of, they're pretty random, right? They're really not specific. They come from a different era. They of course can do great things. They get into tiny spaces that large molecules can't get to. The BBB is one of the main benefits of a small molecule, but you have others, and you have liabilities you have low half-life and low distribution usually because it's so small, it gets filtered. So you got your pros and cons of small molecules, but by far the biggest problem and drawback of small molecules is lack of specificity. And, I used to run all these panels, just screening in random kinases just to see if you might get a hit. And, I've increasingly thought, why don't we do this more in silico? But regardless, it's 20,000 plus proteins. The chances that your small molecule might affect an important protein is non-zero, to say the least. So there's a lot of chemical matter out there. Thankfully, there are certain hints and certain things we've learned over the years of what to avoid metabolically, or maybe from a certain scaffolder chemotype perspective. Some certain problem children in the chemical world seem to come over and over again. But yeah, if you could do a large molecule, I don't see why you wouldn't. There's a lot of people out there who've made so much money in whatever industry, and they just want to invest in something that they think could help people. And GBM's so terrible. I happen to know 3 or 4 people whose family, usually parents, have died of GBM. And it's just such a... But GBM, the victims or the families of the GBM patients often say that, they got a chance. They get 6 months, 12 months to sort of, it's a fatal diagnosis in essence, but there is this sort of small opportunity to spend time with your loved one or not. And I don't know, that's either that's consoling or not, but it's such a brutal disease that has withstood, quite a potent effort against it in pharma, and still has very low overall survival as far as I know. I've been following this company. Dawn is the stock ticker. The company's called Day One. They have a BRAF inhibitor for low-grade glioma, pediatric low-grade. So, but it's you're talking about a couple hundred patients at that point. And we got everyone's favorite boogeyman, which is, you got 200 patients to treat. How are you going to sustain a whole company on that? Well, there is a way. It's called having an extremely high price. And they, interestingly, they priced their drug about half of what I would have expected. And it seems like a very poor decision because if this community of people really is relying on you to advance care, you're going to need capital. And the United Healthcare's and Aetna's, CVS's to pay $400K or $200K or even $1 million for a drug. They do it all the time anyway. So, it's a space that, we've seen this in MS and myeloma. The more money pharma makes, the better the drugs that come out. And glioma and GBM both have been these spaces where nobody's ever made $1. And it's kind of your Pfizer's and Merck's and Novartis's are why should we do it? There's no money here. Let's do something else. And, brain cancer is a real thing. And it's, it's something that deserves more investment. I mean, the pharma industry is just way larger than anything in the med tech or life sciences space. But, for the longest time, these companies did have multiple streams of revenue and businesses. Roche still has what is basically the world's largest diagnostics business inside of it. And then Johnson & Johnson for the longest time had all kinds of things. They divested their diagnostics business a long time ago, but they still have a big medtech business. And occasionally, like Genzyme used to do a lot of diagnoses and different like efforts to get the diagnosis out. I had a drug that had 60 people who took it, and it was such a terrible illness that if you found the patient before onset of illness progressed, you could really save their life. But if you found them too late, it was a permanent sort of neurological irreversible damage. So in that case, like we invested a lot to try to get the word out and try to find these people before it was too late. So I do think drug companies operate in that continuum of like education and diagnostics occasionally, where there's, you know, obviously companion diagnostics for like EGFR and other mutations. Now I think more labs just do them, but back in the day you used to have to do them yourself. Like there's Genentech worked on fish assays for HER2 with partners. So I think, you know, the problem with, as you know, I mean, with brain cancer is you just, you know, have like I said, I think there's a limited investment because there's so few people that have it. So there's just not that much attraction. And this company, day one, I don't think they did anybody any favors by kind of pricing their drug too low, because if they had $1 billion drug, you know, every big pharma would wake up and say, oh, we got to do something in this disease too. And before you know it, the 6-month survival becomes 24-month survival. And that's certainly what happened in oncology. In fact, oncology used to be considered a non-starter in pharma. Now it's actually the largest, you know, revenue sector in pharma. But in the dawn of Taxol, you know, one of the first chemos, and some of the other first chemos like carboplatin and cisplatin, you know, basically they were really poor sellers, and pharma didn't think about pricing back then the way it thinks about it now. And they basically said there's no business here. So few people get cancer compared to diabetes or something like that. So, you know, I think, you know, of course, you know, the extreme, you know, version of this is one patient with one disease gets a drug made just for them. And, you know, that's kind of breaks the whole system we have. You know, we don't have a system equipped for that. But, you know, someday. Yeah, so new FDA commissioner or CBER director, changing p-value. New p-value will be 0.01 going forward. 0.001, actually. Maybe even more zeros will be added by the year. 10-year CARC study required for FDA approval. 2 species. First time ever, 2 species. I'm just joking, of course, but, you know, the market's reacting at least as if medicine's over. And, well, I don't know. Maybe it is.
Hey, Martin.
Hey.
I was just wondering, in terms of developing a new drug, what would you say the biggest price bottleneck or CapEx bottleneck would be? Like, what's the part that requires the most amount of capital? It's not actually developing the drug itself, is it?
Yeah, discovery is pretty simple. It's the clinical trials that cost a lot. So I'm bearish on all the AI drug companies because it doesn't really add up. You know, if you've ever developed a drug, it's the, you know, 5 years of clinical trials you do, and the waiting and the enrolling, and doctors and hospitals and medical centers are very financially driven. If you're going to do a clinical trial, you better pay up, and then we'll do your trial. If you're not going to pay up, well, we got other things to do. And that makes clinical trials really expensive and time consuming.
Okay, so discovery is relatively cheap. So I'm assuming that also includes testing on animal models. It's cheap as well. So the issue is translating it over from animals to humans. What would you say the conversion rate would be between the two in terms of success?
I think that, you know, you sort of take the sort of a staged approach. Like, lots of phase 3s succeed because the company has de-risked it in sort of phase 2. So if you know that, for example, your blood pressure drug lowers blood pressure in phase 2, you can be pretty confident that it'll do so as well in phase 3. But, you know, sometimes you're really rolling the dice. So I think the translation is like, you know, stronger every step you get closer. There's some animal models that translate with high fidelity to humans. There's some that don't. generally anything involving the brain has a pretty poor translation to humans, but a lot of stuff that's like very genetic and metabolic genetics sort of oriented is fairly good translation. Oncology is sort of notoriously poor translation. So, it's a crapshoot, but I think the human clinical trials sometimes cost and take a lot of time because they are very long term. So, that long term nature results in like, let's say you're taking a disease that takes 5 or 10 years to sort of advance. You're going to have to do a 5-year clinical trial, and it's really expensive and it takes a lot of time. Your patent life is ticking away, by the way, as you do this clinical trial. Your patent life's starting to disappear. So if you're looking at Alzheimer's, which is possibly a 10, 20, 30-year disease process, right? How do you do a trial that's longer than your patent life? So I think there's a lot of issues when it comes to medicine that sort of have to get resolved. But the old sort of way of doing things is a little bit, it's not a fantastic system that we have necessarily. So I think the clinical trials are far and away the most.
Right, that makes sense. I agree with you. I would say if during the animal phase, if you can get an effect size that's very, very large, then in that case almost always translates well into clinical. That could maybe remove the need for clinical trials. One more question. I know you're busy. But in terms of drug delivery, in terms of following the science, would you say there are any organs that we currently don't have good delivery mechanisms for, particularly in gene editing?
Yeah, just to take a step back, I don't think there will be a day where we don't do clinical trials. That seems like a really far away.
More so like much smaller scale. Like say it works so well in animals and then you test it on a few humans.
Well, we have that already. I mean, we do have that already. There are many drugs that have gotten approved on a handful of patients, 10 patients, 20 patients. The results are so spectacular that they just get greenlit that fast. But yeah, in terms of delivery, I mean, we have nothing for the lung. The bone is notoriously difficult. The muscle, any kind of skeletal muscle, cardiac muscle are very difficult to deliver. Kidney, bone marrow, these are all very neurological. Anything in the brain as well. Gene editing only seems to work, in fact, for the liver or maybe a local administration of, dermatological or, like a literal direct injection can sometimes work. But in general, systemic gene therapy has failed us, CRISPR and gene therapy, because of targeting. So gene therapy is a little different because you can get those caspids to sort of go to the right place if you try. But certainly on CRISPR, gene editing, we only have liver, and that's much ado about a really big invention in CRISPR, but no actual applications other than liver diseases, which already all these genetic liver diseases. It's a good time to have a genetic liver disease because you got 85 companies all going for the same 3 diseases: Wilson's, ATT or transthyretin, and then what's the other one? The alpha-1 antitrypsin. There's like 3 or 4 rare genetic liver diseases, and they're all being cured by 18 different companies. It's almost gotten hard to develop in that space because the drugs are too good. Like HAE is the other one, and all these companies have like the perfect drug. They're like, my drug's more perfect, and they're all curative basically. So we just love to get something in some other organ, and they do it in hematological, where they just blast the bone marrow and replace the stem cells, which isn't obviously really conducive to great medicine because you are, basically giving a stem cell transplant. So, that's sort of the state of the art. It hasn't changed much in 10 or 15 years, which is sad. And I think there was this hope that AAVs, adeno-associated viruses, would somehow have this tropism where you could engineer, the caspids to actually go to the place that you want it to go. And that doesn't seem to have panned out. There was this huge investment from biopharma VCs and hedge funds to try to get these capsids and different AAV serotypes to go exactly to the right places. And really, we haven't seen anything. The only thing we've seen that's really big is the Avexis drug, which is now 10 years old or so. So, it's been a very, very slow, painful ride. And that's one of the reasons the biotech index, biotech stocks in general, a lot of people are sort of exhausted with a decade of, lost productivity, overcapitalization, all this stuff. And one of the biggest disappointments, I think, is gene therapy didn't carry some of its load that, we lost from other things. And it could have. Same thing with whether it's gene therapy or gene editing. But neither sort of became as promising as they could have been. And then we have all this other bad stuff in the backdrop that cause problems like for starters. No, I'm kidding. But overcapitalization and chasing the same targets was a big one. But I think other parties, whether it's the opioid guys or whoever, didn't do any favors to the industry. But I think, hearing now that the FDA is going to get more conservative is certainly not something the pharma industry wants to hear.
Wait, I'm sorry. So when you say gene therapy, you're referring to like mRNA transient expression, and then gene editing, you're referring to more permanent, like actual DNA editing. And what would you
not
say
exactly
nanoparticles in delivering mRNA? Do you see any hope there?
Yeah. So for gene therapy, I think when we say it in biopharma, what generally we mean is viral concatenameric kind of payload. It's semi-permanent. You get some transfection. It's not uniform. The concatenamers, don't, the episomal, they're not.
Where would that transfection come from if you're delivering just RNA though?
Well, these are viruses, right? So they're AAVs, and they're delivering DNA payloads. So they're basically forming these concatenamers that stay in the cell and get translated, which is surprising. So they don't, for some period of time, there was a thought that they would actually be, be integrated. And, there'd be a lot of concern there with, obviously various issues. The payload for these things is pretty limited in size, but they basically stay as these little episomal constructs in cells, and they read out decent amounts of viral copies. And if you have a disease that's like really, you just need a couple of enzymes to process some big fat, kind of clump, these are perfect. But if it's something where you need tons and tons of protein, you're probably not going to do great with AAV. Now the mRNA is new, right? We have one sort of mRNA modality, which is a vaccine. And certainly the lipo nanoparticles and so forth seem to get you liver expression. But, when I was sitting around thinking about whether the Moderna vaccine would work, I was fairly convinced it would not work because big charged molecules don't just slide into cells, right? They tend to not do that. And, you need to be under 500 Daltons, you need to be nonpolar, you need to, it's all basic kind of chemistry. And, they got it through the LNP where they encapsulate, all this charged stuff inside the LNP. The LNP sneaks in to the liver cell, but it's not sneaking anywhere else in. So I think there's lots of ways to solve this, and I've tried so many myself, and it seems like damaging cell integrity isn't one of them. That's one I tried a couple of times. Like things like anthrax and HIV find ways to deliver payload into cells and keep the cells intact, but they tend to also actually be fairly toxic. So some people have tried to use those. I did a deal with sick kids, University of Toronto, to try to use the anthrax machinery to deliver a valuable payload. AAV is gentler. We get adenovirus infections all the time. We don't even notice. And if we do get them, sometimes if we notice them, we see them as a minor cold. So they're like the perfect way to deliver the payload, but it's just been hard, man. I mean, there's no easy way out of this. And unfortunately, again, this was supposed to be the next chapter of the biotech industry, perfect gene editing or perfect gene delivery, whether it's, like you said, the really super transient RNA or this like semi-permanent, AAV that everybody's using, including Sarepta's AAV, 𝗦𝗡𝗏 with microdystrophin for DMD patients. Or more permanent solution like CRISPR. There's those liver diseases that are being treated, but there's no obvious direction to go next for liver, other than liver. Well, I'm sure some enterprising companies will figure it out someday, and we'll have a biotech boom again, but probably not this decade, unfortunately.
Just to clarify, the AAV, it's only semi-permanent if it's carrying a DNA payload. If it carries RNA, it would be transient. Is that correct?
Well, my understanding is they are DNA viruses, so they're not. I don't.
But if you're not carrying DNA, only RNA, and we're just using the capsid as a way to get
Right.
the actual RNA to the organ, and then it injects it, but injects just purely RNA, then there wouldn't be any concern over permanence.
That is true.
Martin, can I answer this real quick for?
Yeah, go ahead.
Real quick. So the only situation you're describing that leads to permanence is when there's a complementary version of that RNA that binds to it and creates a cDNA, a complementary DNA, which is two single RNA strands together. That would still allow it the molecular machinery potentially to hitch a ride on the types of proteins that transport DNA into mitochondria or to the nucleus. So there is a chance of permanence even with an mRNA administration.
Okay, and is that the concern that people are having about the mRNA vaccines potentially increasing risk for cancer?
Yeah, it's totally possible, but these are super low rates. I mean, it's already a small payload that depends on continued replication of it. And on top of that, to have a statistical chance of DNA formation and transport is another smaller chance, but it's technically possible, and there's a potential for it. And the only other point was about 𝗏𝗏𝗃𝗏. Yeah, you can't genetically engineer embryos because embryos eventually produce germ cells, which means like they produce a sperm or an egg eventually. And if they do that, that means that there's potential of carrying on that gene editing into generations in the future with unintended side effects. So that's why we have what we call the Weissman barrier, or that moratorium in the late 1970s. That isn't like state directed, but like a confederation of states directed, like World Health Organization level. I don't know exactly which organization, but it's an NGO essentially that made scientists agree not to do that. So that's just the other caveat there. All right, thanks. Bye.
Yeah, and when you talk about the barrier, or like I think the point is that the germ cells get the mutation, right? Are you talking about the machinery,
Yes, exactly.
the machinery coming with, or are you more talking about just the propagation of that?
The propagation of that. And the machinery's not going to come with, right?
Right. Okay, just making sure.
Right. But the point is, the propagation of it into haploid cells is definitely possible.
Oh, that's the point. Yeah.
Yeah, that's the key. That's the reason why we have a moratorium on it. That's why he went to jail for it.
But
it's not so much like, hey, these are consenting adults. It's like, bro, you're a scientist. You're supposed to know that you can't do that.
well.
You're bad at your job. You know what I'm trying to say?
Well, just to put.
Like a surgeon operating on your brain and then being like, I'm practicing medicine. It's like, oh, bro, you didn't know about that. Why are you doing that?